Today, viruses are a subject we hear about more and more. We all know the word virus. To most of us, it means a cold or the flu. But what is a virus? Is it a plant? An animal? Is it even alive? For most of us, the only time knowing about viruses becomes important is when we're ill. Usually, a viral infection is just a mild annoyance, a few days of discomfort and lots of fluids. But an infection of herpes virus can mean outbreaks of painful sores again and again for a lifetime. And having this virus flourishing in your blood means a life cut short by AIDS, acquired immune deficiency syndrome. The first thing that must be learned about viruses is just how incredibly small they are. With a light microscope, we can see protists, single-celled plants and animals, fractions of a millimeter long. With the highest power of this microscope, we can just make out another life form, bacteria. These dark specks streaming in the currents. But even smaller than bacteria, and invisible to the light microscope, are the viruses. To see viruses requires this, an electron microscope. An average virus is 1/10,000th of a millimeter in diameter. Turning the magnification up to 50,000 times, they come into view. Dozens and dozens of spherical viruses. Even at this extreme magnification, these viruses look fuzzy and indistinct. To get a clear picture of the structure of a virus requires a special technique called X-ray crystallography. Dr. James Hogle is a researcher in the molecular biology group at Scripps Clinic in California.
There are two major differences between X-ray crystallography and light microscopy. First, a lens cannot be used to refocus the X-ray image. Second, it's impossible to measure the X-ray scattered from a single virus particle.
Since a single virus particle is too small to be visualized directly, the first step in X-ray crystallography is to bring together trillions of virus particles into a workable mass. This can be done by putting a solution of viruses into a small hard plastic container and covering the container opening with a thin tissue called a membrane. The membrane is semi-permeable, which means that the small molecules and ions in the virus solution can move out of the chamber, but the virus particles cannot. The result is that slowly the viruses become less and less soluble, and eventually they form crystals. Each crystal is made of trillions of virus particles packed in symmetric arrays. The regularity of the crystal allows the information on the shape of a single virus to be assessed. To get this information, the virus crystal is placed in a glass tube mounted on an X-ray machine. X-rays aimed at the crystal are scattered into a regular pattern that's recorded on film. This is a picture of that scatter. It is called an X-ray diffraction pattern. Many of these patterns become part of a complex mathematical analysis that produces a kind of three-dimensional map. The map shows the locations of the atoms in the virus structure. With these maps, a knowledge of chemistry, and a bit of intuition, scientists are able to construct a model of a single virus particle. We've learned that typically a virus has two basic parts: a protective outer layer of protein, sometimes coated with a fatty membrane, and an inner core of nucleic acid, either DNA or RNA. Although most viruses consist of only a few thousand molecules, nature has arranged those molecules into a menagerie of shapes. There are viruses that look roughly spherical under the electron microscope, but whose exact geometry is an icosahedron, a shape with 20 triangular faces. There are also rod-shaped viruses. The rod actually has 2 layers wrapped together. At the center is a strand of nucleic acid, while the outside is protein spirally wound in a long helix. Probably the most dramatically shaped viruses are called phage. If you think they look like a spacecraft, you're not alone. The top of a phage is a hollow protein capsule that contains nucleic acid. Beneath that is a shaft called a sheath. And attached to the sheath are leg-like tail fibers. A virus carries only the essentials of existence: a strand of nucleic acid imprinted with the formula for survival, and a strategy to reproduce that precious information, generation after generation. In the complex world of living creatures, the bare minimum simplicity of the virus is striking. Compared to a virus, even a one-celled animal seems like a great factory, performing and coordinating hundreds of tasks. There are cell parts for processing energy, parts for locomotion, and even parts for maintaining the proper balance of fluids. Placing a virus into a protist cell would be something like placing a person into the center of a major city. Both are dwarfed by the large and complex structures that surround them. Without all the machinery of life, how do viruses survive? How do they reproduce? A virus is dependent on a living cell for survival, a host cell. In a way, a virus is a parasite. It attacks a host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle. Interestingly, the lytic cycle does not always happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA. Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates and the destructive lytic cycle begins. Once again, the host cell is destroyed, and a new generation of phage are released to invade new hosts. Plant cells also play host to viruses. Often, the virus is injected through the cell wall by insect carriers, such as aphids and whiteflies. There's no mistaking the vigorous look of a healthy plant, like this melon. But when infected with a virus called zucchini yellow mosaic, leaves become blistered as the virus destroys broad patches of cells. Fruit is also susceptible to viral disease. Here, pepper mottle virus causes discoloration and withering of these bell peppers. Probably the best studied plant virus is this one, tobacco mosaic. Back in the 1890s, it was the first virus to be identified as the cause of a disease. Later, it was also used in classic experiments that would reveal the particle nature of a virus. Animal cells are also invaded by viruses. In fact, viruses are responsible for a large number of diseases, including rabies, hepatitis, and cholera. Some viral diseases can even be transmitted between species. For example, cowpox can be contracted by humans handling diseased herds. The list of human viral diseases is also very long. Yellow fever, influenza, smallpox, herpes, and one of the most highly studied human ailments, polio. The polio virus attacks certain kinds of nerve cells, and for thousands of years, it killed and crippled, especially children. In the spring of 1954, that was about to change. Dr. Jonas Salk and his research team at the University of Pittsburgh developed a polio vaccine. The effort started small, Dr. Salk and his team immunizing a few thousand students in the Pittsburgh area. But soon, it became a nationwide mobilization to protect every child against the polio threat. Salk's vaccine and the vaccine developed later by Albert Sabin were a complete success. And in countries where the vaccines were used, polio all but vanished. No more withered legs, no more withered lives. Today, vaccines play a part in all of our lives. But how does a vaccine work? It takes advantage of the body's own natural defense against infection, called immunity. When a virus or a foreign bacteria enter the body, special blood cells called phagocytes seek out and destroy the invader. This picture shows phagocytes colored in red in pursuit of several rod-shaped bacteria colored in green. As a second line of defense, molecules called antibodies also are produced in response to an infection. Antibodies are highly specific. The protein shell of a virus triggers the production of a particular type of antibody. In turn, the antibody is effective only against that particular virus. Antibodies attach themselves to the protein shell of the virus and render the virus inactive and make it easier for the phagocyte to finish the virus off. What a vaccine does is stimulate antibody production, and in many cases, a vaccine is nothing more than a solution of inactive viruses. Polio viruses for polio vaccine, smallpox viruses for smallpox vaccine. The first step in making a vaccine is isolating the virus you want to control. This is done by applying viruses to a culture of host cells. After a while, clear patches in the culture appear, showing where cells have been destroyed, which is also the location of the virus. Antibodies are effective only if there is an exact match at the antigenic site, the white spots on the virus shell. If these sites change, antibodies cannot attach, and our immune system cannot protect us.
Some genetic mutations in viruses alter their surface structure such that they're no longer recognized by antibodies. This kind of mutation is responsible for periodic epidemics of some viral diseases, such as influenza.
Because a disease virus can have many different varieties or be very changeable, it is sometimes impossible to develop a vaccine against it. A quick change virus is hard to stop, but there are some things even worse. Imagine a virus whose target is the very immune system cells that defend against infection. That nightmare has become real, and it is called the AIDS virus. Human immunodeficiency virus, HIV for short. They are engulfed by phagocytes, but they are not destroyed, and they go on to attack the cells that produce antibodies, leaving the body defenseless. HIV is one of those viruses that does not go into its cell-destroying lytic cycle right away. In fact, the nucleic acid of HIV can reside in our blood cells for years, causing no discomfort, no symptoms. The only way to see it is with a blood test. That fact makes HIV a dangerous epidemic threat. Medical research to cure or control AIDS is working along several directions, from drug therapy to vaccines. But the most powerful defense against HIV or any disease virus is understanding it. Remember, a virus is just a parasite, and like any parasite, it must be passed from host to host. From what we know now, the AIDS virus is transmitted sexually, as well as in blood and blood products. Stop the transmission, and you stop the virus. Although viruses have always been a part of our world, we have still managed to survive. One step at a time, we have learned about viral disease, and one step at a time, we are learning how to stop it.